folks, welcome to Anatomy and Physiology on Demand. This is lecture 11B, part 4, where we're going to be talking about synaptic transmission. We're going to be talking about different types of synapses. So as we have mentioned before, some neurotransmitters are excitatory and some are inhibitory, and we're going to look at various ways that neurotransmitters cause their activity. We're not going to be exhaustive by any means, but we're going to basically look at one each of various different type of categories and then talk about what happens at the end to the neurotransmitters. Okay, so some excitatory, some inhibitory, some it depends on the type of receptor that's on the postsynaptic cell, some open ligand ion channels, some are going to act through second messengers. So, and some is going to be fast and some is going to be slow. So as we go through these various types, this is what you need to figure out. Is this excitatory or inhibitory? Is this ligand gated or second messenger? And is this fast or is this slow? Those are the three questions for you to ask yourself about each of these. So the first one we want to talk about is a cholinergic synapse. So as you recall, a cholinergic synapse is one where acetylcholine is the neurotransmitter. And the only type of cholinergic synapse we've talked about is in the skeletal muscle, and that is one where acetylcholine is excitatory. And we have spent a lot of time talking about this cholinergic synapse, so I'm not really going to go over it anymore. I think we all have mastered it. If not, please come see me either in virtual office hours or one-on-one, or send me some emails if you don't understand this process, or watch some videos on it. I do want to point out that in some cells, acetylcholine is a neurotransmitter, but it causes an inhibition, and this is seen in cardiac muscle as opposed to skeletal muscle. This type of neurotransmitter response is fast. It only takes a few milliseconds. The next type of neurotransmitter synapse involves a neurotransmitter called gamma aminobutyric acid, which you get to call GABA. Okay. And this was one of the ones that I mentioned earlier was super important in creating memories, and GABA and glutamine had some import to when it came to Alzheimer's beyond the scope of this class. So the way GABA works is by the same mechanism as acetylcholine. In other words, it's going to bind to a ligand receptor acetyl ion channel, and it's going to cause a change in the membrane potential. The ion channel is going to open and same release. But as opposed to acetylcholine, remember what kind of ion did acetylcholine let enter the muscle cell? Okay, in contrast to that, the GABA receptor is not on that type of an ion channel, but is on a chloride ion channel, meaning it's going to allow chloride movement. And I also want to point out the acetylcholine ion had a positive charge, but chloride has a negative charge. And so here is an example showing what happens. When GABA binds to that chloride channel seen on that orangey postsynaptic cell, that allows chloride with its negative charge to enter that cell. So if this is a postsynaptic neuron, that means negative charges are entering that postsynaptic neuron, which means the resting potential inside that axon is becoming more negative or is hyperpolarizing. The net result of this, a stronger impulse would be needed for that postsynaptic neuron to reach its threshold. Okay, so hopefully you can see how this, even though it works the same mechanism as acetylcholine, because it's a different ligand, it's causing an opposite effect in a postsynaptic neuron. This also is very fast, only taking a few milliseconds. At this point, I want you to pause and take a second and review how G protein acted as a second messenger, because that's going to be important for a type of synapse we're going to talk about shortly. Okay, now that you've had a chance to review that, I want to talk about a compound known as adrenaline, which I think all of you have heard about before. So I don't want you to memorize this chemical stuff on the left, but just look at it for a second. We start with amino acid, and on the benzene ring, we change a hydrogen molecule to a hydroxyl, and we change it to a different amino acid. Or we could start with the second amino acid. Then the next thing we do is we add another oxygen, making another hydroxide component, and that gives us a compound called L-DOPA, which then we can convert into dopamine. And so L-DOPA and dopamine is something I'm going to talk about when we get to the nervous system next week. I just want to point out these two compounds now so that when I mention those words, you will realize, oh, they are precursors to adrenaline. I understand this concept now. So after dopamine, we are going to end up with another hydroxyl group, which gives us something called noradrenaline, which is the same thing as norepinephrine. It's like the Greek versus the Latin. And then finally, if we would add a methyl group to norepinephrine slash noradrenaline, you would end up with epinephrine slash adrenaline. So at this point, I don't really care if you call it epinephrine or adrenaline, just stick with whichever term you are most comfortable with, and realize their abbreviations are going to be different. If you don't know either, I would suggest sticking with adrenaline because that is the word that is the root for this type of synapse. And that type of synapse is called an adrenergic synapse. See the word adrenaline, A-D-R-E-N, adrenaline in there. And an adrenergic synapse is excitatory, and basically, we call it adrenergic. However, I'm going to be talking about noradrenaline, which can be abbreviated as NA, also known as norepinephrine, which is abbreviated as NE, is acting as a transmitter. And this is also one that can be excitatory in skeletal muscle and inhibitory in cardiac muscle. But this one is very slow by comparison. It takes multiple seconds, not milliseconds, multiple seconds to minutes to happen. And that's because adrenergic synapses involve a G protein. So looking at the activity of norepinephrine, in this drawing, norepinephrine is binding to its receptor. When it binds to its receptor, there's a G protein, peripheral protein on the inside of that cell, that one of the subunits breaks off and then binds to that transmembrane protein, adenylate cyclase. Hopefully this is ringing some bells from way back when. And adenylate cyclase is the enzyme that allows ATP to be converted into cyclic AMP. So cyclic AMP is the second messenger system. And as you recall, cyclic AMP activates protein kinases. So we all know that kinases do phosphorylation. So yes, it can phosphorylate a protein, But in addition to that, it also can open ion channels, some calcium channels, not the slow ones, and some sodium channels, and also can have something to do with transcription. Okay. So for us, it's not important to understand exactly how it's happening. I just want you to realize that adrenergic receptors are working through a second messenger system involving the G protein going to cyclic AMP going to protein kinases. So after we have had our synapse, we're going to stop our signal. So one of three things, just like we talked about before, I'm just going to go into a little bit more detail and put it all on one slide. The first option is the neurotransmitter is kicked off of its receptor and it's broken down while it's still in the synaptic cleft, and those fragments will have no effect on anything. So this is what happens with acetylcholine. When it's kicked off the acetylcholine receptor, there's acetylcholine esterase there in the synaptic cleft. It breaks it down into the choline and the acetate. Neither one of those can bind to anything. The second thing is the reuptake. So either the neurotransmitter itself or one of its fragments can be reabsorbed into the presynaptic cell. So we've already learned about this with acetylcholine, where the choline part of acetylcholine undergoes endocytosis and is now in a can be reattached to acetyl-CoA to form acetylcholine inside a new synaptic vesicle. As far as other neurotransmitters, the actual amino acid itself can undergo exocytosis whole, and then there would be a different enzyme in that presynaptic terminal bouton. A classic example is a monoamine oxidase, and it would break down the neurotransmitter in the presynaptic cell itself, so it could not be reused. And the third option is the neurotransmitter and or its fragments will gradually diffuse away. And so the classic example is if we go to the acetate component of acetylcholine, it diffuses to areas of lower concentration in the extracellular fluid. Okay. So here is a GWAP showing pre and postsynaptic cells and things labeled 1, 2, 3A, 3B, 3C. So talk yourself through this process, pause it here, and once you have written everything down, and I would actually suggest writing it down to make sure you have it, then continuing on, because I did post answers on the very next slide. So don't go ahead until you have already written down your answers. And there they are. And with that, we are done with lecture 11B in its entirety. And I want to thank you all for continuing to work so hard, and I will see you during the next lecture session if you want to join in for the Zoom meeting if you have any questions.